Antibody generators, also known as antigens, are any foreign thing that enters the body. Most antigens are harmless, like peanuts or pollen, but some can be very dangerous, like the flu or Ebola virus. But harmful or not, our bodies are programmed to defend against these. So when one enters our body, there are two main responses that can happen. The first initial response is the innate immune system. This is involved in the creation of interferon proteins and lymphocytes. Interferon proteins, as their name suggests, interfere with viral reproduction, and lymphocytes are white blood cells that help fight off infection. Sometimes this is enough to halt the further spread of infection, but other times you need the adaptive immune system. This has two main categories, the humoral and cell-mediated response. The humoral response happens when the virus is outside of the cell, but if it has infected the cell, you get the cell-mediated response. This creates T cells which target and kill the infected cells.
Cells routinely chop up their proteins and place the pieces on an MHC1 complex that is then presented on the outside of the cell. If the cell has been infected or become cancerous, it will have abnormal proteins present. And if a T cell finds that piece of abnormal protein, it sounds the alarm for invaders and differentiates into either a memory cell or effector T cell. The effector T cell binds to the infected cell and kills it by either creating holes in its cell membrane or by making it commit apoptosis, which is programmed cell death. Imagine a T cell is a policeman and is checking IDs in a lineup. If it finds that one of the suspected criminals, or in this case infected cells, has an abnormal protein for its ID, the policeman would throw the infected cell into jail, or in this case, kill the cell.
Both parts of the adaptive immune system lead to the production of memory cells. Memory cells are made when B cells produce antibodies for an invading virus. That way, when the same virus reappears, the memory cells made from the previous infection will be able to recognize it and produce the correct antibodies much quicker. This is how vaccines work, except instead of infecting you with a live strain of the virus, a dead or weakened one is used.
In the production of memory cells, you can think of the B cells as policemen and the virus as a criminal. So the B cells use the antigens from the virus as the fingerprints that they store in their database. So when the virus infects again, they know exactly how to defend against it.
But as harmful as viruses are, they can actually be used as life-saving devices. In several studies, reprogrammed viruses have been shown to target and kill cancerous cells in mice. The process uses a well-known and studied virus, such as the adenovirus, which is then manipulated to target certain proteins that only cancer cells have. The virus is then injected into the patient where it continues to spread to the rest of the cancer. Cancer cells are often undetectable by your immune system, so this process is able to shine a light on the cancer so your immune system is able to spot and kill it. So as you can see, viruses were and still continue to be deadly to mankind, but now we're able to use them to save lives. As this technique improves, hopefully it will become more accurate and widely available. I'm Olivia Feeney, and thanks for watching my video.